Hello, bio buddies.
Hello, bio buddies.
I'm Dorian,
And I'm Ross.
and today we're going to talk to you about sex. But not what you think, actually, about meiosis. Meiosis, in fact, is a reduction division of a diploid cell containing two n numbers of chromosomes to form four haploid cells containing n number of chromosomes. Now, before we go into interphase, I'd just like to say that at this time, the cell is performing its normal metabolic activities. That means it's growing, it's feeding itself, it's creating organelles, it's breaking down organelles, it's excreting things it doesn't need and taking things it needs. And so, cell's pretty normal right now, but someone had sex. So, the cell must create four gametes. And now Ross will tell you the pre-phase of meiosis.
That's really interesting, mate.
No problem.
Right, so I'll start an interphase. The cell replicates chromosomes here, and each chromosome has two sister chromatids.
Now, to illustrate this, I think we should go to our little makeshift whiteboard that we have here. If you bear with us for a second. Now, I know you can't see much because of the light reflecting. I'll quickly move up and change that. Right. Sorry about little disturbance. As you can see on the graph paper, we have the cell. And the plastic bag illustrates the cell nuclear membrane. Underneath, just to give you a little peek, we have four chromosomes. Now, at this stage, they are in the form of chromatin, unidentifiable scramble of little lines that you would see in a microscope. There we go. And at this moment we have four different chromosomes here, as you can see. And during interphase, as Ross so expertly described, these homologous pairs of chromosomes, as you can see, pair up. Yeah, does that make sense? Good. And during interphase, let me just remove the cell nucleus. Don't think it's not there anymore. Haha. To form a replicated homologous pair of chromosomes. There's one.
Very well illustrated, if I may say so, Dorian.
Thank you, Ross. I always appreciate it. Bear with me one second. And here we have that replicated stage that is completed by the cell during interphase. Of course, this is all under the cover of the cell nucleus. During prophase, which I will now describe to you, these initial homologous pairs replicate to form replicated homologous pairs. And also, once I take off this cover, again, don't believe it's gone, these replicated homologous pairs exchange DNA, which is one very important characteristics of meiosis. And they do this at points called chiasmata along the sister chromatids, and here I will illustrate. So this piece of DNA jumped over to land on this chromatid. As you can see here in detail, we have crossing over occurring. The same thing happens here. And of course on the larger replicated homologous pairs here. Whoops. Bingo. Now this is all occurring underneath the nuclear membrane. However, during this phase, as spindle fibers and centrioles appear on either side of the cell, as I'm illustrating right here, the nuclear membrane also starts to dissolve and disappear. And now it's gone. So now these crossed over replicated homologous pairs are now drifting aimlessly in the cell, and also tetrads are formed as clusters of four chromatids here and here. And now Ross will explain to you metaphase.
Alright, I'll get into it. Metaphase. I'll explain metaphase. Metaphase, the centromeres of each homologous pair of chromosomes are attached to a spindle fiber. The spindle fiber's attachment is to ensure that each chromosome or homologous pair of chromosomes moves to opposite poles of the cell. They line up at the equator to do so.
Got it. I got it. There we go. Rough illustration, but you can get the point. Oh, thank you for that, Ross. Now, we move to anaphase 1. These homologous pairs are now separated from each other, from the tetrad, like so. As you can see, there's a clear separation occurring in the middle. And they are subsequently pulled by the spindle to each end, also called the pole of the cell. Now, as you can see, the chromosome number is halved, but each chromosome still consists of 2 sister chromatids. At this time, the cell membrane starts to indent. This signifies that it is getting ready for its cell membrane to divide. And this happens clearly in telophase 1, which Ross will explain right now.
Telophase 1. Alright, so here the cytoplasm divides through cytokinesis. Each chromosome, or pair of chromosomes, homologous pair of chromosomes, found at opposite poles, and the nuclear membrane reforms around each daughter nucleus.
Now let's imagine that we have 2 plastic bags signifying 2 nuclear membranes. Alright, at this time I'm going to have to rub out the spindle and the centrioles,
so that we can start meiosis 2. But just before we start meiosis 2, I'd like to add in a bit of information about interphases. There's a brief interphase stage here, which however does not include the S phase of the original interphase stage, so it's just a brief interphase stage before we go into meiosis 2. Isn't that right, Dorian?
During prophase 2, the spindle fibers reform and the centrioles reform in each of the 2 new cells, here and here, and these attach to the centromere. Excuse my little alteration of speech there. Now, we will go on to Ross, and he will tell us about metaphase 2.
Metaphase 2, not 1, not 2, it's even better than metaphase 1. Right, so the chromosomes line up at the equator of the cell, and the spindle microtubules attach to the centromeres of each chromosome. Now, if that is not exciting, I don't know what is.
Alright, thank you for that. Back to me, anaphase 2. The spindle microtubules, right, they pull the sister chromatids apart. Can you imagine the force needed for that? So the sister chromatids pull apart to each pole, right, like this, right, creating a clear distinction in the middle, right? As you can see, there's a clear separation occurring in the middle. And we're in anaphase 2, so the spindle fibers, they're pulling these sister chromatids apart to each pole. Each one wants their own share of the chromatids. I can't blame them. The chromatids now become chromosomes. And that's the interesting part I'd like you to remember. Okay, back to Ross. Telophase 2.
Telophase 2. Right, nuclear membrane reforms around the 4 sets of daughter chromosomes. Now the cytoplasm divides through cytokinesis. And as a result, we have 4 haploid sex cells with n number of chromosomes present. And just to add some information on top of that, there's a thing called non-disjunction during meiosis that can occur, and that is just when chromosomes do not separate normally, and this can lead to a number of different problems and a number of different disorders. But we won't get into that because we'd need to make a new video on that. So that's it from us today.
That's coming up in the next video.
Next video.
Be prepared for that.
Depends how successful we are on this, if we get the sponsorship deals and whatnot, but yeah, we'll see how it goes.
So I hope you enjoyed our video on meiosis. Remember, it's another version of sex that I'm sure you haven't heard of. Alright, more from us in next week's episode of Biology IB. Alright,
Let's go
keep
for it.
enjoying the IB. Don't let it pull you down, and have a great day. Bye.
Bye.